Hello everyone, welcome to another lecture of Fundamentals of Computational Biology. So, this is the last lecture of this course and we will again revise some of the very important sections that we have learnt so far. So, today's topic is revision of examples of biological processes and their modeling approaches. So, let us begin the lecture. So, the examples of biological process and modeling approaches, we have learned a lot of these examples in our previous lectures, but why I choose to revise this particular section today is because researchers or PhD scholars can grasp a lot of examples and how these processes are being governed in the regular normal cells. So, they can actually learn how mathematics and computational biology approach help them to identify the problem and help them to identify the exact solutions associated with those problems. Now, biological processes are intricate, complex series of actions that occur within a living organism. Think of them as the inner working of life itself. These processes enable organism to grow, reproduce, respond to their environment and so much more. They are like instructions that is governed at the very aspect of life. Now, what is their role inside a living organism? Now, why these processes are so crucial? Well, they are the driving force behind the life's most important fundamental activities, from the beating of your heart to the way plant photosynthesize. All the living organisms rely on these processes to function and survive. Biological processes encompasses a wide range of activities such as DNA replication, protein synthesis, metabolism and cellular signaling. They are like the gears and circuits that keeps the biological machinery running smoothly. As we explore these processes today, we will also learn how computational biology helps us to model and understand them in a better way. So, get ready to learn these particular processes step by step. The first topic that I want to cover today, it is about the genetic regulation. So, in genetic regulation, we will first look at the transcriptional regulation part. Now, genetic regulation is like conductor of an orchestra, ensuring the genes are expressed at the right time and the right amount. One of the primary ways that happens through is through transcriptional regulation. Now, transcriptional regulation is a process by which genes DNA sequence is used as template to create a complementary RNA molecules. Imagine you want to open a tap for regular water uses. Now, the amount of water needed, that should be dictated by the regulation of the valve. So, what will happen is, as long as you open the tap of your valve, you open the valve of your tap, the amount of water you will receive. So, this particular regulation is required in a genetic level or gene level also. Now, let us move to the next slide to explore another aspect of genetic regulation, that is called as post transcriptional regulations. Post transcriptional regulation is like a musical piece that can be further redefined and interpreted during the performance. Gene expression can undergo additional regulation after the transcription. This is known as post transcriptional regulation. You might have studied all this in your UG courses. So, think of in this way: you have transcribed the musical score or genes, but how it can be played can vary. Post transcriptional regulation is like adding the dynamics, tempo changes and the individual interpretation to the performances. Post transcriptional regulations include processes like RNA splicing, RNA editing and the control of RNA stability. The process can change the composition of RNA molecules and it can be produced during the transcription. Ultimately, these influences which proteins are made from the RNA. One fascinating aspect of post transcriptional regulation is how it can fine tune the gene expression rapidly in response to specific stimulus. So, it is like making a quick adjustment to the musical performance to match the mood of audience. Together, transcriptional and post transcriptional regulations are essential components of genetic control, allowing organism to orchestrate their performances to a constantly changing environment. In the following slides, we will delve into the how computational biology helps us models to understand these intricate processes in a better way. So, we will also look at the modeling of genetic regulations. So, let us see what are all different mathematical model for gene expression. Let us start. Now that we have explored the genetic regulation, let us delve into the computational biology segment where we will use mathematical models to identify the gene expression levels. Imagine you have a piece of sheet music and you want to predict the precisely how it will sound when it will be played in an orchestra. Mathematical models in a computational biology are like musical notations that precisely describe how genes are turned on or off, how RNA is transcribed and translated, how proteins are produced and then being folded. For example, one mathematical model that is widely used is Michaelis Mental equation. You might have seen it in enzymatic driven chemical reaction. It describes the rate of enzyme catalysis reaction. It shows how enzyme facilitates the conversion of substrate into a product. This equation allows us to predict the rate of gene expressions based upon the factors like concentration of molecules, the binding of proteins to DNA and the timing event in the cell. Now, let us see some of the basic concepts related to regulatory network modeling. So, regulatory network modeling is like a map of all the musical instruments in the orchestra, showing how they interact and influence each other. In biological systems, genes does not work in isolations. They are part of a complex network where one gene expression can impact the expression of another genes. So, regulatory networks models helps us to visualize the intricate connections. For instance, consider a network where gene number A, B and C are intact. The mathematical model can represent how changes in the expression of gene A might affect the gene B and gene C. These models use graphs and nodes to illustrate these interactions, providing the insights about how changes in one part of the network can ripple through the entire system, affecting various biological processes. In the following slides, we will explore the specific examples of how these modeling approaches have been used so far, including mathematical examples to gain the insights into the biological processes. Now, let us talk about metabolic pathways and how they are helpful in case of computational biology. So, now let us talk about what are those metabolisms. So, welcome to the fascinating world of metabolic pathways. In this section, we will explore how computational biology helps us unravel the intricate dance of molecules within our cell. Now, imagine metabolism as energy producing choreography of our cell. Just like the dancers follow a sequence of steps, our cells follow specific biochemical pathways to convert nutrient whatever we are consuming, to convert them into energy. This involves a lot of synthesis, a lot of chemical catalysis pathway in the entire cycle. Metabolism is a complex network of chemical reactions, with each reaction catalyzed by the enzymes. Computational biology allows us to model these reactions mathematically, just like the choreographers design dancers in a routine. For instance, the Michaelis-Menten equation helps us to predict the enzyme kinetics, that how different substrates influences the rate of reactions. These equations are like the choreographer's instructions, telling us how fast a dancer should move, or their movements depends upon the which factors. Now, what are the metabolic pathway analysis? Let us dive deeper into the metabolic pathway analysis. Think of it as studying the choreography of a dance performance to understand the individual moves. Metabolic pathway analysis involves mapping out all the chemical reactions that occur inside a cell, from the intake of any nutrient to the production of energy and the synthesis of essential molecule. Is like creating a detailed dance routine from start to finish, ensuring every step to occur in a exact particular time. Computational tools helps us to analyze these pathways, identifying the key reactions, intermediates and the bottlenecks. We can quantify the flow of molecules and energy within the pathway. This is really very crucial for understanding how cells functions and respond to the change in the environment. For instance, the steady state assumptions is a mathematical concept used to describe the balance of metabolites concentrations within the pathway. It is like ensuring the dancers to maintain their positions during the dance performance. Now, another aspect is constraint based models. So, now that we want to predict how changes in the environment can affect the metabolism, or how it can optimize in the, it can be optimized for a specific purpose like biofuel production or drug discovery. That is where the constraint based models comes into the picture. Imagine you have a dance routine, but you want to adjust it how different stages or the spaces can be used. So, constraint based models allows us to make these adjustments in context of biological pathways. This approach uses mathematical constraint to represent the limit or boundaries of chemical reactions. It is like the telling the exact molecule how they can perform and what are the parameters within which they have to perform. For example, if you have to produce a biofuel, let us say there are 10 different molecules which are involved in this entire pathway. So, experimentally we can identify the ranges of these parameters, and we can actually make a constraint based approach where we will define the functionality of the exact bioreactions and the ranges within which all these molecules have to work, and together using this constraint based approach we can actually know what will be the concentration at the end of these reactions, and based upon the output you can actually predict the exact amount of concentration that you required in order to produce or in order to meet the given conditions. Now, one common example of constraint based modeling is flux balance analysis, or also called as FBA, which optimizes the metabolic pathway to maximize the production of desired product. It is like finding the best dancers in a routine to showcase the most impressive moves within the performance. Now, another field where this approach is really helpful is the cellular signaling. So, welcome to the signaling cellular world. In these slides we will uncovered the intricate communication systems within the cells and how the computational biology helps us to decode their secrets. So, let us talk about the overview of signaling. So, it is like imagine the cellular signaling as a vast communication network, similar to how we send the messages or signals from one to another. In the world of cells, signaling is how they talk to coordinate the various functions. Now, cellular signaling involves the molecules like hormones, neurotransmitters and various proteins that conveys messages from one particular part to another part. Think of these molecules as messengers delivering notes between different parts of the cells. They trigger the responses like cellular growth, immune responses or gene expressions. Computational biology provides us with the mathematical models to understand how these signals are transmitted and interpreted within the cells. The one common model is the receptor ligand binding equations, which helps us to predict how molecules bind to the cells. Now, receptors and the initial stage signaling cascades. This equation is like a mathematical playbook that explains how messages or in the form of ligands interact with the receptors to produce a cellular response. Now, what are these signaling pathway models? So, let us explore the signaling pathway models further. Picture these models as maps detailing the routes and stops of a complex transportation system. Signaling pathways are consequences of events where the signaling are transmitted from cells to the surface of nucleus, instructing the cells to respond for that particular situations. Computational models of signaling pathway helps us to visualize and predict how these events occur. For instance, we use ordinary differential equations, also abbreviated as ODEs, to describe how concentration of signaling molecules changes over the time as signals propagate through the pathway. These equations are like equations that describe how traffic follows through a transportation networks, helping us to predict where and when and what might occur within the cells. Signaling pathway models also allows us to study the effect of perturbations such as mutations or drug interventions on signal transmission. They are like simulation tools that helps us to understand what happens when we tweak this particular transportation system. This allows us to predict the importance or the impact of disruptions on the cellular behavior. Let us talk about the network analysis in a cellular signaling pathway. So, moving on to the network signaling or the network analysis in cellular signaling, imagine this is examining the entire communication infrastructure of a city. In cellular signaling, it is about the studying how different signaling pathways and molecules are interconnected. So, network analysis uses mathematical concepts like graph theories to represent and analyze the complex cellular communication networks. Now, you must have noticed the concepts of ODE, the concepts of graph theory or the vector spaces that we have already learned in our previous lectures. So, let us continue the lecture. These networks shows the interactions between the signaling molecules and helps us to identify the critical hubs and the pathways. It is like creating a city map with different routes and intersections. With computational tools, we can predict how disruptions in one part of the signaling network affects the other pathway. For example, if we want to block a particular signaling molecule, the metabolic network analysis can help us to anticipate the ripple effect through these networks. Much like predicting the traffic congestions when a road is closed. In summary, the cellular signaling is like a vast communication network within the cell, and the computational biology equips us with the mathematical models such as receptor ligand binding equations and ordinary differential equation, as well as the network analysis tools like graph theories to decipher its language and implications. In the upcoming slide, we will explore the real world example, and we will see deeper how these systems affect the entire biological systems. Now, we will look at the population genetics model. So, let us start with basics of population genetics. Now, welcome to the world of population genetics, where we will explore how genetic traits are passed down from one generation to another generation. In the upcoming slides, we will uncover the fundamental principle and the mathematical models behind this intriguing field. Imagine we are studying a group of friends with different eye color, trying to understand how these eye colors are inherited and changes over the direction where the generation. So, that is what the population genetics does on a larger scale with the entire population. So, population genetics deals with the genetic variations within and between the population. It helps us to answer the questions like how do genetic traits spread, how do the new traits emerged, and how does all these can be visualized using mathematics. So, one fundamental concept is allele frequency, which is like counting how many people in the group or friends have blue eyes. So, this frequency changes over the time due to processes like genetic drift and natural selection. So, we will explore these part in our next slide. So, let us talk about the genetic drift and Moran model. Now, let us delve deeper into the mathematical models behind the genetic drift. Think of these models as tools that helps us to understand how the eyes of color of our friend changes over the time. Genetic drift is like random shuffling of a deck of cards. So, it is a process where a certain genetic traits become more or less common in the population purely by chances. For example, if only a few friends have green eyes, their green color might become more or less prevalent in the group over the generations. One widely used mathematical model to understand genetic drift is Moran model. This model simulates how genetic variation changes within a population due to random effects. It is like playing a game where we randomly select the individuals and decide whether they pass on their traits to the next generation or not. The Moran model helps us to visualize the chances events that allelic frequencies in a population changes over the time. On one hand, the natural selection is like the friends with the most appealing eye color or getting more attention. It is a process where certain traits become more common because they provide an advantage for the survival and reproduction. For instance, if a blue eyed friend are more attractive to the potential partners, so the frequency of blue eye in the population might increase over the time. So, models like Wright-Fisher models helps us to understand how genetic variation is maintained or disrupted by natural selection. Both genetic drift and natural selection are crucial drivers of evolution. Computational biology allows us to simulate these processes and understand how genetic diversity within a population changes over the time. Let us talk about a case study now. So, the case study is about the evolutionary dynamics. In this slides, we will embark a journey into the exciting world of evolutionary dynamics, we will explore with the help of a detailed example of how computational biology uses mathematical tools to understand the ever changing the tapestry of life. So, let us see this example. Imagine we are stepping back in time, observing the evolution of population of birds with different beak sizes, and these are located on a remote island. So, this is like our detailed example of evolutionary modeling. So, the evolutionary dynamics studies how genetic traits changes within the population over the time, and in our case it is like over the generation. It is like witnessing a grand symphony of life that is being unfold itself. In our case study, we will focus on how beak sizes in a bird evolved in response to the environmental changes, particularly the availability of food. Now, so one commonly used model is Wright-Fisher's model, which we mentioned earlier in the context of population genetics. This model helps us to simulate how genetic variation within a population changes over the time due to genetic drift and natural selection. In our bird population, we can use the Wright-Fisher's model to predict how distribution of beak sizes changes as the birds with different beaks sizes interact, reproduce and pass their traits to the offspring. So, we can also incorporate factors like mutations and environmental pressures such as food availability into the model. The mathematical equation in the Wright-Fisher model allows us to make predictions about the future of bird populations. We will see how beak sizes adapt to the changing environment. Will certain the break size becomes to more picture into as advantageous for the survival and reproduction, that we can predict within the with the help of this example. This case study provides a practical example of how computational biology can help us to answer these questions. The Wright-Fisher model is a mathematical model widely used in the population genetics to describe how genetic variations within a population changes over the generation due to the genetic drift. This model is named after the after the scientists Sewall Wright and Ronald A. Fisher, who contributed significantly in this particular field of computational biology, or more precisely population genetics. So, I will explain the Wright-Fisher's model here. So, Wright-Fisher's model uses the following assumptions. One is the population size capital N is constant and it does not change from one generation to the next. Another assumption is the individuals in a population reproduce to produce in a next generation, and the offsprings are randomly sampled from the parent generations. Now, the mutations are rare and have negligible effect on the allele frequency within a single generation. Another assumption is the population is non-overlapping, meaning there is no migration or the individuals from in and out of this particular populations. So, the mathematical equation that describes the Wright-Fisher model is relatively straightforward. It calculates the probability of observing a specific allele frequency in the next generation based upon the current allelic frequency. So, this probability follows a binomial distribution. So, I will shortly explain the Wright-Fisher's equation, that is P of q dash equals to how n chose over the k multiplied by q to the power k multiplied by 1 minus q to the power n minus k. So, let us explain what are these factors in this equation. P over the q dash is the probability of observing an allele frequency q dash in the next generation or upcoming generation, where n is the total population size. K is the number of copies of alleles of gene of interest in the current generation, where Q is the frequency of allele of interest in the current generation. In practical terms, researcher uses these equations to predict how frequencies of different alleles in a population changes from one generation to the next generation due to random sampling of individuals in the reproductive processes such as genetic drifts. So, in context of our bird population and their beak sizes, the Wright-Fisher model would be used to simulate how beak sizes alleles frequency changes over the generation in response to factor like food availability and the selective pressures. This helps researchers to understand whether a certain beak sizes become more advantageous for survival reproduction in the ever changing environment. By studying the detailed example of evolutionary modeling, we can gain insights into how mathematical models can shed lights on the mechanism of evolution itself and helps us to understand how species adapt to their surroundings. In the upcoming slides, we will explore the more real life word example, and we will continue to unravel the mysteries of computational biology. Now, let us talk about very briefly talk about systems biology. Welcome to the realm of systems biology, where we explore the intricate web of we will see the intricate web of interactions within the living organism. In these slides, we will delve into the principles and methods of system biology, including how it utilizes the mathematical modeling to understand the complexity of life. Systems biology overview is where we begin our lecture. Think of system biology as viewing the life through a wide angle lenses. Instead of focusing on individual genes or proteins, it considers how all the components in the biological system work together, much like examining how the various parts of a car interact to make it function. Systems biology aims to uncover the underlying principle governing the biological systems by examining how molecules, cells, proteins are interacted to govern the functionality of a particular cell or the entire organism. This holistic approach helps us to understand how disease develop, how organisms respond to their environmental changes, and how to engineer the biological systems for various applications. Mathematical models play a vital role in systems biology, allowing us to simulate and predict the behavior of complex biological processes. These models helps us to understand how various components interacts, how signals are transmitted, and how a system respond to the perturbations. So, in this section we have already covered the network metabolic part. So, now here is the application part. We already know how one particular gene affect the expression of various other genes. So, if we have already information about the functionality of these genes and the regulatory part of these genes, now we can engineer the system itself. So, for example, we do not want any regulation in my gene from part A. What I would do is, I would select a system where this regulation part is missing, and it works in a way that it will be expressed during the all time. In biology, this condition is also termed as the constitutive expression, and you must have studied all this in your UG classes, that how you can choose in with the help of genetic engineering, how you can choose a part where a particular gene is constitutively expressed. So, you copy the part where the regulation part is the regulation sequence are present, and you put it to the gene of your interest where you want the constitutive expression. So, genetic engineering in a practical way can be visualized using the concepts which we have learned so far in case of computational biology. So, this is all the recap we can collect from the portion of the biological processes example. I wish all the best to the learners, so that they can utilize all these concepts in their research work. Also, this will be helpful to generate a new insight, it will open a new form of platform for the learners who are restricted in a biological term. They can learn how to predict these systems, they can also learn how you can model and produce a lot of forecasting in this particular field. So, I wish all the very best, and thank you very much for attending this particular course. Stay connected, and you can ask whatever doubt you have, whatever doubt you might have within this particular course. Thank you very much.